In Lecture 8 of the course, you're going to learn how to find and analyze publicly available RNA-seq data. And to do this, you're going to pull that data directly into the R bioconductor workflow that you've been developing throughout the course thus far. All right, so let's just review our learning objectives. Today, you'll learn how to use command line tools to reach out and grab raw FASTQ files directly from large public repositories like the Sequence Read Archive and download them to your computer. You're also going to learn about something called the HDF5 file format or hierarchical data format. This file type allows us to store large amounts of data, really of any kind, along with associated metadata. You'll then explore the ArchS4 database programmatically, and this ArchS4 database we're going to talk in a lot more detail about is actually structured as an HDF5 file. And then finally, you'll start and finish the Step 4 script today, which is available for download here and on the scripts tab of the website. And that Step 4 script is really not that substantial. It doesn't involve downloading any new R packages. Everything in terms of the R packages will be familiar to you already. And it's not, frankly, that many lines of code and not that many unique things that we're doing. Okay, so the last and certainly not least is that there's something additional you need to download because we're not going to work in the Step 4 script with our course data, right? So that's a big difference compared to previous lectures. After all, this lecture is about public data, so the Step 4 script really demonstrates how to work with any data set. And so you'll need to download the ArchS4 database in this HDF5 file format. And I've provided those to you on Dropbox. Here's a link to the human and a link to the mouse. Just a word of warning, those are sizable files. Each is about 7 gigs in size. So each of these files is about the size of 2 FASTQ files that you downloaded at the beginning of the course. But they contain a heck of a lot of data. So each of these files contains over 230,000 samples worth of RNA-seq data. And so how we can store that much data in a single file and only have it be 7 gigs, and how we then tap into that file and navigate our way around those 250,000 samples to find what we want is really the subject of that Step 4 script. So if you want to download both of these collectively, you'll have over 500,000 samples worth of RNA-seq data at your disposal on your laptop. Now, for the purposes of today's lecture, we only need the mouse data. So if you have concerns about hard drive space, you just need to download the mouse. Give yourself plenty of time to download it. It just could take a while to download to your laptop. Okay, so only download the mouse data for the purposes of today. All right, so really, I think the first thing to address is when on earth would you need to access public RNA-seq data? And there's at least 2 scenarios you can envision. And the first is probably the simplest. And that's that you've read some cool paper and they publish an RNA-seq data set and their analysis of it, but you want to look at that data set from a different angle. Maybe it complements a study you're doing and you want to now compare your own data with this published data set. So I'm going to assume that data is publicly available. That's oftentimes a prerequisite of publication now, that authors deposit their data to well-known data repositories like GEO, SRA, or ENA, and we'll talk a bit more about these in just a minute. But what's less clear sometimes is whether or the extent to which you'll be able to match the data, the FASTQ files, with the metadata that describes the samples from which that RNA-seq data was generated. Right? This metadata is exactly what we have for the course data set in our study design file. So let's assume you can either get that from GEO, SRA, or ENA, one of these data repositories, because they provide mechanisms for authors to upload metadata, or that you can infer it from the paper. Okay? If these 3 things are all a go, then the only thing really standing between you and doing the analysis is you just need to get the FASTQ files, and you need to just download them from SRA. Sounds really simple, right? You're good to go. It's often not as simple as you would think. And this has to do partly with the way SRA is organized and some of the user interface and user experience aspects of working with SRA as a website. So we'll talk about solutions to deal with that. The second scenario is maybe less pleasant, which is you need to get large numbers of samples or multiple studies that involves lots and lots of FASTQ files and finding a place to download and store those. and then trying to process them all through the same pipeline so that that's uniform. And so that's a much more challenging scenario, but we're going to talk about solutions for both. All right, so the strategy, at least for addressing that first scenario, is really simple. It's we access that public data, download it, and we have those FASTQ files, and then we carry them through this workflow that you've already been working on. The strategy for the second scenario is different because the problems are different, and that involves actually skipping download of raw FASTQ files altogether and instead tapping into a different kind or source of that data where it's already been aligned for you, and it's been aligned for you actually using Callisto. And so what you download is not the sizable raw FASTQ files, but already summarized read counts from a Callisto output, and we can just pull that right into a different step in our workflow and move right through those first two step scripts that we've already discussed. And we'll do all that in the step 4 script today. Okay, so if we're going to talk about public sequence data, we have to talk about the Sequence Read Archive, or SRA. It is the NIH's, NCBI's main mechanism by which people make their data publicly available, and not just for RNA-seq, but any high throughput sequencing application. So it is a vast database. And so maybe before we talk about how to get data out of SRA, we talk about just how to get data in, because oftentimes that'll be the first thing you'll probably be faced with is you have some data you're about to publish and you need to make that data available. And the best way to do that is through SRA, right? Wrong. The best way, in my opinion, to get data on SRA is to go through GEO. GEO is another database, also through NCBI. Unlike SRA, which is all things sequencing related, GEO is gene expression. It's the Gene Expression Omnibus, GEO. And so if we're talking about RNA-seq data and we need to get that RNA-seq data into SRA, you should be aware that if you deposit it to GEO, GEO will broker your data to SRA on your behalf, meaning your data will be available on both sites, on GEO and on SRA. And that's advantageous, at least in my opinion, because the process for submitting data to GEO is much more straightforward. So if you're interested in that process and a sort of step-by-step protocol, I just point you toward our lab protocols site again, where we have under RNA-seq data analysis, submitting data to GEO, what you need to do to get ready, how to complete your metadata spreadsheet, prepare your data for transfer, and then transfer it. All right, so that's one way to think about, at least in the short term, clearing that hurdle of how you get your data out there. All right, so what about getting data out of SRA? Well, again, my approach might sound a bit counterintuitive, but it's that you don't actually have to use SRA to do that, right? SRA is an NCBI resource, but there are other resources, right? Not everybody deposits their data to SRA. There's also the European equivalent of that, which is the European Nucleotide Archive, which is under EMBL and EBI. And the reality is these two agencies talk to each other, right? And so data that's on SRA is kind of mirrored in many cases or available on ENA, and vice versa. Data on ENA is available, so is available on SRA. So if somebody deposited in Europe, if somebody did a large-scale RNA-seq study, or a small one for that matter, and deposited their data to ENA, it oftentimes appears on SRA. So that's why I say to get data out of SRA, you don't need to use SRA. My personal preference, again, it's just my opinion, I'm not trying to offend anyone because both of these groups are fantastic and a lot of resources go into developing and maintaining all of these sites. But my preference is for ENA. And I think, again, I feel like this comes down to user interface and user experience and web tools. So if you're going to use a website to click and point and download files, FASTQ files, I think your best bet is probably going to be with ENA. All right, so this is what the ENA site looks like. It's very clean. It's got a search box. You can search text or for accession numbers as well. So I'd like to, at this point, navigate to that website. And you can click on this link if you want to go there as well. And so here I am in my browser. I'm just going to zoom in a little bit, make this a little bigger. And I'm also going to just go over to RStudio where I've printed off my study design file for the course data, just so I can copy and paste a SRA accession number. So if you were reading a paper and you got these SRA accession numbers from the paper, you could just enter it really anywhere. I'm going to enter it in the text field here and say search. Even though it's an accession number, it'll probably turn up if I search it in the text field. It's one of the reasons I like ENA. It just works. And sure enough, here it is. This is the data actually from Camilla's study. We didn't deposit it to ENA. We didn't deposit it to SRA. We deposited it to Geo, and it sort of percolated or cascaded through to SRA and ENA. So if I click on this run, that's the number I entered right there, I can read about that run, human, RNA-seq, skin, healthy sample number 1, should look familiar. And right here, I can see that there are read files available. So I'm going to show those. And in just a second, those read files will display here. And sometimes this takes a minute to show up. There it is. This is the single sample. I could now just click this box and download the selected files. Done. All right, now this was just one sample. It's easy in the search page to search for more than one, but that's it. That's all that's involved in getting these files from ENA. And again, we never deposited them to ENA, but that's the value of having these agencies talking to each other. Okay, so that's how I recommend if you're going to use a web interface for getting data out. But I also want to point out that several RNA-seq aligners are actually SRA, what I call it SRA aware. I don't know what the real term is, but what I mean by that is you don't actually even need to download the data from SRA. You can point the aligner to the data on SRA, and it'll complete that process of pulling that data and running the alignments. Unfortunately, Callisto is not one of those. Neither is Salmon, as far as I'm aware, the two main pseudo aligners that we've talked about in class. But both HiSat2 and Bowtie2, two really well-known aligners, will both do this. And they do this if you provide a single parameter here, --SRA accession, ACC. That then tells HiSat2 and tells Bowtie2 that what it's about to see is an SRA accession number, and it should do the alignment directly from that accession number. So this is possible because of a whole toolkit that NCBI created called the NGS toolkit, which you could read more about at this GitHub site. That's what's happening under the hood here. It's going and getting this data and downloading it and doing the alignment. I don't use HiSat2 and Bowtie2 much at all, at least not for RNA-seq data. So I can't say I've had a lot of experience with exactly how these work in practice, but the fact that it's even possible is really great, especially if you're using one of these aligners. All right, so what about if the point and click's not going to cut it and you're not using Bowtie2 or HiSat? Are there other command line approaches for getting data out of these repositories? And the answer is yes. And the main one you're going to hear about is SRA tools. Again, GitHub sites down here. And again, it's from NCBI itself. And they have a very popular function called fastqdump, which will just, if you provide it with a SRA accession, it will just go and get that fastq file and download it to your computer. That fastqdump, which you still see referenced a lot, has largely been replaced or improved upon by faster qdump, which does all the same stuff but does it better, faster, etc. And so it's really simple to use. Once you have it installed, which you can read about here, you just say faster qdump, the name of the accession number for the file, and then if you have multiple threads on your machine, you can use the -e parameter and tell it how many threads. Remember, cores and threads, we've talked about these before. Why -e and not -t? Because they reserve the -t parameter for something else with faster qdump that we don't need to get into. So again, you don't have this on your computer now. You don't need it. We're not going to use it in the class. This is me helping you be aware of what's out there. All right, so the problem with some of these command line approaches is they don't get out the metadata. So right, here's our metadata for our course data set. So although faster qdump SRR, et cetera, will pull out this first sample, this healthy sample 0, 1, it will not tell us that it's healthy. And so this is that point I alluded to just a minute ago, that being able to sort of match your data with the metadata is oftentimes the problem. So how do we get our metadata out of SRA? And one really, I think, interesting solution, and one that's from Penn, actually from Rick Bushman's lab when Lewis Taylor was in the lab, is called GrabSeq. And GrabSeq actually works with the SRA tools under the hood, but it provides in my opinion, a more convenient interface and way of interacting, not just with SRA, but with several databases. And so once, if you install this, and we're not going to use it in the course, this is just letting you know it's available. If you install GrabSeq, and you just type the program and then tell it you want to search SRA. In this case, -T refers to the threads, so 4 threads. You can then tell it you want not just the data, but you want metadata available for that sample. Okay, so program, where to search. Other options are MGRAST and iMicrobe. Those are both databases that have a lot of sequence data for microbiome and metagenomic and genome sequencing studies. Number of threads. You tell it you want to capture the metadata -M in this file, and you give it the accession. And you can check that out on his GitHub site as well. Okay, but like, what happened to being lightweight and portable, right? Nothing I've said so far, if you really think about it, is that lightweight, right? Waiting for a FASTQ file or 10 or 100 to download is not fast, not lightweight, and frankly, the latter, downloading 100 FASTQ files, is not likely to happen on your laptop. Certainly not the same laptop that we just did our Callisto alignments on. So, what do we do, right? What if we have that second scenario and we need to get a lot of data? And I've already told you, if you think way back to the first lecture, that the amount of data out there is increasing at an incredible pace due to the democratization of sequencing, large and small sequencers being increasingly available. So how do we really tap into that and access that data in a way that's efficient and lightweight? So the first thing I would just highlight is that this is a significant challenge and one that is not going to improve. So as an example, this is Geo's site as of today, and you can actually see the number of experiments that they have of data available for. And I've highlighted what these series entries look like. This is actually one from our lab. And the idea initially, early on in the days of Geo and early on in the transition from microarray to RNA-seq, is that as data was being deposited to Geo, the team there that will load that data into Geo was going to process it in a way that you would have this Geo data browser that would allow you to actually start to look at the data, make a heat map, slice different parts of that heat map, etc. But that became really an untenable process. I mean, the deposits to Geo skyrocketed, and the number of samples or entries or experiments that were manually curated to produce these kinds of analytic interfaces by the Geo staff were really not keeping pace. And that's because these data repositories really are only able to efficiently act as data repositories, not as a systems biology analytic platform. So that means the burden of analyzing this data is probably going to fall on you as the investigator. You're going to have to pull this data out of a repository and analyze it. So one of the solutions to that, or one of the things that works to our advantage that I just want to reference, we're not going to use this data in the class per se, but you'll find out how to access it, is the idea that there have been a number of community-based projects or consortia projects that have generated tremendous amounts of data related to a particular theme. One of those is the Genotype Tissue Expression, or GTEX project, and the GTEX project is a public resource from cadavers of healthy tissue where they have RNA-seq data from non-diseased tissue sites, liver, heart, brain, and others, paired with oftentimes whole genome or whole exome sequencing and samples from those same individuals biobanked, right? So an incredible repository. And it allows researchers to link genotype, right, from the whole genome sequencing and whole exome sequencing with tissue expression from the RNA-seq data. And so that GTEx project is one of the data resources you could go to, and they of course have a portal, which you could see here, and you can access or interact with some of the data there, but we'll take a look at a strategy for, again, programmatically accessing that today. In a similar way, the Cancer Genome Atlas, TCGA, involves a tremendous amount of data stratified by obviously different types of cancers and their tissue of origin, also oftentimes paired with lots of orthologous data sets like whole genome sequencing or metabolomics. And the portal for that you can see here. We'll also see how to access TCGA data. And then finally, ENCODE, which has really multi-dimensional data sets across the omics spectrum, has a ton of RNA-seq data from cell lines, from single cell data sets, from tissue, from whole organisms. It's in fact the second largest sort of category of assays that make up ENCODE. And so there's a whole lot of these sort of community-driven projects, and in large part, they've really helped to standardize sample collection, storage, and data generation. However, the tools for then carrying out rapid and large-scale analysis of these community-driven projects have really lagged behind. And that's probably to be expected. I mean, necessity is the mother of invention. Now that we have these large data sets, now is the time, and many people have capitalized on this to really build some of these tools for large-scale analysis. So what are those tools? Well, those tools have been evolving for a long time. This is by no means a need that has not been recognized until recently. In fact, as far back as 2011, Jeff Leake's group had developed Recount, which at the time was impressive, right? 475 samples of RNA-seq, 8 billion reads, 18 different published studies. This is really one of the first, attempts to gather up RNA-seq data in one convenient R accessible location, right? So Recount was an R package. That was followed up about 5 or 6 years later with Recount 2. It's a major update to the Recount package, really major. So now over 70,000 samples instead of 475 across many more studies that came from SRA, GTEx, TCGA, you name it. Many of the community projects I just mentioned now bundled inside this single R package that you could access, all right? So the main premise here was, let's process this data. We'll use computationally, resource-rich approaches to process all this RNA-seq data ourselves, Recount, and then we'll make available via Bioconductor the tables of read counts so that you can just start your workflow with those. That's the idea. It's a great idea, very straightforward. So what next, right? Has it moved beyond Recount 2? Well, to discuss where this has gone, I just want you to recall, some text I put up when we were talking about Callisto that really highlights some of the main advantages of Callisto. And this is from Leor Pachter's blog, that the implication for biologists with these pseudo-aligners like Callisto, like Salmon, is that now instead of freezing an analysis, you can explore your data dynamically, quantify against different transcriptomes, re-quantify. You can really analyze data locally on your laptop instead of using more computationally intensive means, which means the analysis is portable, right? So there's a few key phrases here: explore dynamically, analyze data locally, analysis is portable. And that really then enables the kind of studies that allow us to go in and tap into huge amounts of data, right? So we used Callisto and valued it because it allowed us to do a small, analyze a small data set on our laptop. But imagine if you had substantial compute resources, what could you do with something like Callisto? Well, that's really what was addressed by Avi Mayan's lab, which was, all right, now if we scale the resources and still use a really efficient tool like Callisto, we should be able to align massive amounts of publicly available RNA-seq data from the main organisms that people work with, human and mouse. All right, and this is called ArchS4, which stands for All RNA-seq and ChIP-seq Sample Sequence Search, right? So an ambitious project to profile all RNA-seq and ChIP-seq data. And the workflow here that Avi's lab used is not too complex. It's really similar to what we've already done, but now scaled to operate on a server with many more resources available. So data was retrieved using SRA tools and FastQDump, actually, which you now understand, to get the FastQ files. Callisto alignments were carried out. Gene level counts were then embedded or stored in an HDF5, which is often abbreviated H5 structure. Okay. They also did some TSNE embedding and created a really useful website that we'll explore in just a minute. But that data, they had to make a decision about the extent or how broad they would be in what they captured. They largely pulled data from human and mouse, analyzed on either a HiSeq series sequencer or a NextSeq. All right. So if you're looking for an experiment from human or mouse sequenced on one of these machines, chances are it's in ArchS4. We can actually take a look at this database website in just a minute. A lot of the information I just told you is here, including links to the paper. I also linked to the paper in today's reading for the lecture page. And you can see the breakdown of the study. But if you click on download, the first thing I want to show you is if you click on download, you'll see the exact files that I offer you on Dropbox, right? They're here. They're about 7 gigs each, human and mouse, okay? You'll also see that they have all the data from GCTX. They have the data from Recount2, right? So a number of different data sources here. If we go back to the main page and we click on Get Started, there's this pretty incredible TSNE plot that we'll display in a minute of all the data in ArchS4. And one of the things that you can do that I encourage you to kind of use as like a query tool is you can search for, within this large TSNE network, you can search by tissue type or cell line to find exactly the samples and experiments you want. Those will display down here and they can be easily downloaded. And we can just come back to this in a minute. Okay, so that's how ArchS4 works. So the great thing about this is because you use a super efficient pseudo aligner, the cost per sample has now dropped to less than a penny, right? So this now becomes sustainable, right? You could scale this and continue to develop it. Currently, as I showed you a minute ago, it has about 250,000 mouse samples and more human samples. Okay. ArchS4 is not alone in this endeavor. Another group has also embarked on a similar process. Their paper is published a little bit later in GigaScience, but the idea is similar. Reach out to SRA and pull data using FastQDump. Once you get these FastQ files, you can do some QC work on them to get filtered FastQs, because if you're working with filtered data, it's going to run more quickly. And then they run STAR and Callisto on this data to produce the information, the counts, which can then be accessed on their website. And this is the Digital Expression Explorer 2, D2, but can also be accessed via an R package. So both ArchS4 and D2 are available and accessible through R. Let me just see if that finished displaying. Yeah, so you get this TSNE here that you can rotate around. It's pretty incredible. If I'm looking for, CNS, let's say astrocyte gene expression, That will highlight on the plot. Doesn't look like there's that much astrocyte data. So maybe we'll try something immune related, myeloid, THP1s. And if this is enough data, I can't even see it here. You could select it. It'll show you the data here and you could download it. All right. It's right there. I don't know if you can see that. There's some data points right there. All right. Anyways, that's really all I wanted to illustrate from ArchS4. The rest of it we'll deal with in R. Okay, if you're interested, D2 is evolving quickly. This was in 2019. They had about 229,000 samples of RNA-seq data completed for human and about 250 for mouse. Now it's 357 for human and 545 for mouse, so an incredible amount of data. And they have even more queued up and ready to go. So that blue is queued. They're also not just human and mouse. They do have other species as well, but much less data. Okay, so how do we then make this lightweight and portable? All right, I didn't actually tell you how we'll do that. And the way we'll do that is by taking advantage of this HDF5 file format. So hierarchical data format, which is called HDF5, but you may also see it referred to as H5, is often called a self-describing file format. So what does that mean? Well, it means that each of the elements within the HDF5 file have annotations or metadata associated with them. And you can think of an HDF5 file as really a directory inside a file, so a file system inside a file. But instead of folders and files, HDF5 files are made up of groups and data sets. So groups would be analogous to a folder. Data sets would be analogous to a file. So in that way, conceptually, very, very similar to a file system. Each of these things, the groups and the data sets, can have metadata attached to them. And even the HDF5 file itself can have metadata attached. So this is perfect for storing gene expression data. So why is it great? Well, not only is it this file system contained in a single file, but it can store huge amounts of data, which we'll talk about in just a minute, keeps the metadata with the data, doesn't care what kind of data you have or if the same data types are stored within the same file. So you could have completely different types of data from different assays in the same HDF5. Easy to share because it's a single file, independent of your operating system or platform. And importantly for us, as we'll see today, even if the HDF5 file has a lot of data in it, we don't need to read the whole file into memory to actually work with it. We can access just the groups or data sets that we want. Okay, so I won't go into great detail about this, but there's a lot of other uses of HDF5. So understanding how to access these in R and what they are is going to be broadly useful outside of bulk RNA-seq. For example, the well-known biome file that's a common data structure for microbiome data is, at least the version 2 biome file that everybody uses now, is an HDF5 file format. Single cell sequencing initiatives, 10x genomics, the .loom file, those are HDF5 files as well. Oftentimes large RNA-seq data initiatives, are packages from, for example, the 10x brain data, so single cell data of brain samples. It's all sort of represented as an HDF5. RNA-seq data, we talked, or you might recall, when we talked about Callisto being able to do this bootstrapping, that it stores those bootstraps in a .h5, which is an HDF5. And then finally, high throughput screening, like the kind the Broad Institute does in their clue.io project that has over 1 million different data sets from different perturbations. That .gctx file is actually an HDF5. I told you it could store huge amounts of data, and although we like to think of biology as being big data, it just isn't compared to other fields, particularly physics and astronomy, where, for example, the LOFAR telescope, so the low frequency array telescope, that's an ensemble of telescope setups across Europe. This is just one of them shown here. I think this one's in Ireland. They produce huge amounts of data on the order of 35 to 40 to 50 terabytes of data per hour. And that all gets structured inside HDF5 files. So these are really many, many images of extremely high resolution. The IceCube Neutrino Observatory in Antarctica also has a huge data collection initiative. They have 5,000 sensors across a kilometer of ice looking for these neutrinos. And that's all structured as an HDF5 file as well. So really good for truly big data. All right, so today we're not dealing with neutrinos, we're not dealing with LOFAR telescopes, but we are going to deal with our favorite topic, RNA-seq data. And so we're going to work with a particular data set, one that I helped contribute to, that came out of Igor Brodsky lab, and Naomi Phillip, who actually took one of the early iterations of this course and has a figure from our course in this paper. We're going to talk about this data, and we're going to see as a test case if we can, treat this as our cool paper that we want to access the data for, and then go out and get the data from that paper. So the goal for today is, can we find this data set in ArchS4? Is the metadata readily available? Again, I find this to be the most challenging part of accessing public data. And then finally, can we go from data to analysis really rapidly in R? Okay, so that'll be the goal for today. We'll take a break right now, and for the second part of the lecture, we're going to focus entirely on the step 4 script.